to all. I am very glad to invite you all this international FDP program. I am Dr. R. Krishnaveni, HOD, PG and Research Department of Microbiology, Idhaya College for Women, Kumbakonam. And I am convener and organizing chair for this 5 days international FDP on biological and industrial applications. A warm, pleasant welcome to you all. I am in chance of my happiness. I am privileged to welcome our principal sister, Reverend Sister Eugene Amala. My gratitude welcome to you, sister. And I welcome our secretary, Mother Reverend Sister Claudine Aroke Mary. Then I welcome administrator, Reverend Sister Anthony Amal, and vice principal, Reverend Sister Vimali Mary. Welcome you all, sisters. My hearty welcome to staff members from various colleges and universities. Now I am happy to welcome today's speaker, the senior research scientist, vice principal, R&D, Dr. K. Sendilkannan from EGS Pillai Arts and Science College, Nagapattinam, India. My happiest welcome to you, sir. Now I hand over this session to Nithya ma'am.
Thank you, ma'am. Good afternoon to everyone. I am Dr. M. Nithya, Assistant Professor in Microbiology Department. Sharon Breenet says that start each day with gratitude, practice kindness, and end it with a peaceful heart, and watch your days become a little bit sweeter. Let us start our first day of International Faculty Development Program on Biological and Industrial Applications, organized by PG and Research Department of Microbiology, Idhaya College for Women, Kumbakonam. Let me first of all start by giving glory to the Almighty God for making today's occasion sound success. Michael Bolani says, personal participation is the universal principle of knowing. I feel privileged to welcome all the participants from various colleges, universities, and those from other countries. For the seamless conduct of the virtual seminar, all the attendees are kept on mute mode. Participants, I request you can post your questions and doubts in the chat box during the session, and after conclusion of the talk, an expert will answer all your queries. An investment in knowledge always pays the best interest. Idhaya College for Women was established in the year 2000 in Kumbakonam. The aim of this college was to provide quality education to women and to encourage their growth and development in all sectors. The institution also aims at all-round development of the students and not just concentrates on academic excellence. Owing to this, the institution trains the students to imbibe cultural and ethical values. The institution with the motto "Strive to Shine" has been able to produce many successful academicians, scientists, administrators, and many more who have contributed excellently to their fields and the society as well. The college has a strength of 3,250 students pursuing 10 undergraduates, 8 postgraduates, and 7 research programs, including 1 doctoral degree in Tamil. Also, our college shifted to conducting online webinars, FDP, with programs with the students in various departments. The faculty members are available online during interactive sessions for students to clear their doubts. And PG and Research Department of Microbiology was upgraded with sophisticated equipment and laboratory facilities. The thrust areas of research in the department include organic farming, vermicomposting, mushroom cultivation, and horticulture. Students of our department have excellent in both academic as well as extracurricular activities. They have assured 33 university ranks in undergraduate, 22 ranks in postgraduate, and assured 7 gold medals, including myself. Faculties in our department have published several research articles in both national and international journals. Don't talk, act. Don't say, show. And don't promise, prove. I deem it a great honor and privilege for me to welcome our college principal, Reverend Sister Amala, who builds characters, inspires dreams, encourages creativity, builds confidence, instills a love of learning, touches our hearts, and changes our lives forever. Now she is going to deliver an inaugural address of this program.
Cheerful wishes to all. I am extremely delighted to note that the Department of Microbiology, Idhaya College for Women, Kumbakonam, is organizing an online international FDP program on biological and industrial applications. Science and everyday life cannot and should not be separated. The brain is the citadel of the senses. This guides the principle of thought. Scientist is someone who observes, experiments, measures, explores, predicts, invents, and wonders. With many research activities now taking on a global dimension, it is imperative to discuss positive approaches towards inculcating best research integrity practices, including examining the role of the academic publications in setting the standards for integrity. This FDP brings together relevant field experts, professors, clinicians, industry representatives, postdoctoral fellows, and research students from around the world, providing them with an opportunity to discuss scientific questions, achievements, issues, and challenges in the field. Organizing such an event at this point of time reinforces our objective of developing an environment for the exchange of ideas towards technological developments. This FDP would be able to deliberate on current issues of national and international relevance. I convey my warm greetings and felicitations to the organizing committee for their laudable effort in conducting this FDP and also the participants. I am looking forward to the unique opportunity to learn and network with professionals in this international gathering to learn about the latest technology. Let us have an exciting and memorable event filled with enlightening interactive sessions. We continue our ascent towards success by asking more of ourselves today, tomorrow, and forever. May God bless us all.
Thank you, sister. Next, I would like to invite our department HOD, convener, organizing chair, Dr. R. Krishnaveni ma'am, to introduce our resource person, Dr. K. Sendilkannan sir.
The senior research scientist, Dr. K. Sendilkannan, obtained his BSc degree in physics from Trivika College at Thiruvarur, MSc Physics from AVVM Sri Pushpam College at Pondi, Tanjavur district, MPhil in MS University, Tirunelveli. He has passed the UGC SLET examination. He has completed MBA, PGDCA, B.Ed, CA, MA, DCA, and DCH, and also awarded DLit, Doctorate of Letters, for his academic and research provisions and achievements. He is having 21 years of teaching experience. He has published more than 300 papers in international journals, UGC CAD Listed, Springer, Elsevier, Scopus, Web of Science. He has presented 300 papers in national and international level conferences, seminars during his academic career. He has presented more than 100 papers in conferences, seminar, mainly invited talk at MK University Madurai in February 2012. His publications are ultrasonics, spectroscopy, material science, crystal growth, crystallography, nanomaterials, biomaterials. Dr. K. Sendilkannan got so many awards. Best Dean R&D Award India 2018. Best Vice Principal Award India 2018. Best Indian Educationalist Award India 2018. Best Academician Award in India 2018. Highest Research Paper Publication Award for 2018 he received in February 2019. Received Global Scientist Award Malaysia 2018. Received Senior Scientist from Ministry of Commerce Industries at 2019. I am very happy and proud to introduce you, sir. Now I hand over the session to Dr. Sendilkannan, sir. Sendi,
Yeah,
sir.
thank you. Yeah, ma'am. Thank you.
Sir, now the session is
Yeah, ma'am.
yours. Shall I share the screen, sir?
Yeah, ma'am. Yes, ma'am. You can go to the first slide, ma'am. I will start the session.
Okay, sir. Okay, sir.
Yeah, good evening to one and all. Before the start, I would like to thank my management, our management from Nagapattinam, EGSP Group, for giving me this opportunity to participate in the international FDP. Also, I would like to thank the management of Idhaya College, the principal, the head of the department, Dr. Krishnavani ma'am, and all the team members for this international FDP program. And I am very glad that I am one of the resource persons in this particular FDP. So today, my talk as a R&D man and physics related to the bio applications, bio, pharma, and industrial applications of crystalline material and nanocrystalline materials. This is the topic. So I'm the R&D in charge here in our college. Our college is affiliated with Bharatdas University, and we are having UGC 12F and 12B, and it is a NAC A-graded institution, and we are waiting for autonomous status. The process are over. So I am starting with the slide number 4. This is crystal system. So there are, before entering into the bio or pharma applications, what are the materials we have to grow so that it will lead to some bio or industrial applications. So when we are having the application in industry or electronic side, this is one of the setups. When you are having bio applications like pharma, bio, that other setup like physiotherapist like that, then we'll have other applications. That is bio applications that we can confirm. So the crystal system generally having 7 types: cubic, tetragonal, orthorhombic, monoclinic, triclinic, trigonal or hexagonal, rhombohedral, like that. So the 7 types of crystal system having 14 types of Bravais lattices. So before growing the crystal, these are the parameters we have to know what are the bond length and bond angles for the particular 7 types of systems. So these are the general crystal systems. If anyone is growing the crystal, the system will adapt with these 7 types, any one of the 7 types of the system, which is said to be already available. So the new crystal will be of any of the 7 types: a cubic, tetragonal, orthorhombic, monoclinic, triclinic, trigonal, rhombohedral, hexagonal, like that. Now, spectroscopy. So we have to analyze the particular specimen, a bio specimen or an organic specimen for bio application, like that, for which we are in need of spectral tools to identify the peaks, identify the wavelength, wave numbers, like that. So the particular spectroscopy having the absorption spectroscopy or emission spectroscopy, it may be elastic, it may be reflection, impedance, inelastic, and coherent, like that. I'll just give one example for each to identify the particular spectral applications. Absorption, so some energy is said to be absorbed so that we will have some variation in the energy as well as the light output. Emission, so it is having the excess energy that is said to be released as a photons, that is a emission. Elastic, it can be over and it may regained the value of momentum, like that. And inelastic, it will not get regaining. And reflection, it is part of the representation to and fro motion, like that. And impedance, it is an effective value of AC resistance. Coherent, it is an amplitude and phase, like that we have to measure for each wave. So the crystalline materials are macro crystals, micro and nano crystals. These are the 3 types of main representation of crystals. So whenever we are going the crystals, there are 2 types of crystal specifications are there, importance are there. One is non-linear optical crystals, NLO crystals. Other is non-NLO crystals, or they are call it as centrosymmetric crystals. So this we have to grow according to the solution growth method or any other type of methods. We are having more than 10 methods are there, including SR method for bulk crystals. A human being height nearly 5 to 6 feet, that height we will grow the crystals like that. For that much of height, we can grow the crystal by SR method. For tiny crystals, for the lab purpose and academic purpose, we can go for solution growth method. And non-linear optics, the dielectric polarization varies with the electric field. This is the non-linearity. there we have one limit that is called Swinger limit. Based on that, we can identify that it is a NLO crystal. Second order NLO means we'll have green light emission. Third order NLO means we will have 1/3 of the what is the wavelength given like that. So if it is the 1000, we'll get 300. It will be in invisible region. So from the invisible as well as from green light, we can identify the second order or third order representation of the particular crystal can be identified. So if we are having a bio tool or any other material, fluorescence will help in identifying the particular specimen is said to be properly grown without any flaws. So that is the slide number 12 represents the fluorescence. Fluorescence is nothing but when you are having the input energy that is said to be given, the output energy is from our material. So that will be in the pure visible region. So input wavelength will be some 300 nanometers range. The output will be more than 300. It may be 400, 350, 400, 450 like that. So this property is called refranging property. Refranging means there is a change in the wavelength. This is the fluorescence representation. So next slide number 13 represents resonance fluorescence. So resonance fluorescence is when the input and output are having same value, when the excitation and the emission are same, then it's called resonance fluorescence. This is the general representation. And next is luminescence. So many types of luminescence are there. Here it represents the cold body emission. Our material will have either fluorescence or luminescence like that. Slide number 16 represents fluorescence versus luminescence differences. See, fluorescence is a type of luminescence. It has the absorption of photons, and fluorescence is taken place in atoms or molecules. See, what we are taking from the chemistry or physics lab, we are having only the atoms or molecules. When you are going for organism, bioorganism like microbiology, biochemistry like that, we have to go for luminescence applications. Luminescence is for organisms, solution, and higher molecules. Fluorescence for atoms or molecules for lower level, especially for the purpose of crystals and nanomaterials. Crystalline solids growth and preparation method. Generally, crystals are grown by solution growth method. Solution growth is we are taking a beaker, the input of 2 or more reactants set to be kept over there for a period of minimum 10 days, a maximum of 1 year, or within 1 or 2 months. That also period we will take according to the specimen what we have taken the chemical composition. So within 30 days we will have the seeds, small crystalline with dust is set to be formed. Using butter paper, we can clean the crystal and we can go for the single XRD, single crystal X-ray diffraction pattern to identify the ABC parameters as well as the alpha, beta, gamma parameters, so that we can identify what we have grown, the same material we are identifying, the glycine malate or the amino and pyrans as well as benzopyrans like that we are having. The same is said to be grown over there or not, we can identify. So this is single XRD available at IIT Madras. Solution growth method is the easiest method and cheapest method. And when you are going for nanocrystals, directly we cannot go for nanocrystals or nano representation of materials directly. For nanomaterials from zinc oxide or other oxides, we can grow directly. For nanocrystals, we have the crystals, crush the crystals by ball milling method. A milling balls rotating over there for a period of 40 hours like that, more than 1 day. So when the crystals with a bulk shape, that can be converted into next level of nanoscale by ball milling of 40 hours continuous rotation over there. So their morphology is said to be somewhat differs. Here, the current and the future perspectives over this nanobiotechnology is we are going for one structure. So many people they are asking FDP as well as conferences. So we are having good samples. How we can identify the particular sample is having some bio or pharma applications, and how we can conclude that they are having some industrial application in electronics and all. So before entering into that, the slide number 21 represent one chemical structure, C8H11NO2. So based on this compound, when the compound structure according to the chemistry, the chemistry department people, they know very well, when the structure is very straight, that with the hydrogen, huge hydrogen and all, they will have good optical properties first. Second one, they are having some beauty product, cosmetics, beauty products like that, that application they are having more. When the chain is said to be somewhat broken somewhere, or they are having side chain, like benzene ring with ortho, meta, para, we are having sulfur or oxide or any other types like that, then they can have some specific applications like pyruvates and anti-cancer and other application, anti-diabetic, anti-cancer, anti-inflammatory applications like that. So continuous chain and branched chain, they are having the difference. Now this structure, what we are seeing in the particular PPT 22, they are having the same C8H11NO2 structure. So they are having C8H11NO2, but their position and structure differs. Chemical formula is same. Physical representation in the application of the diagram as well as chemical combination of the particular oxygen in the meta, ortho and para situation is said to be differs. It's all 8 materials. They belong to C8H11NO2 only, the formula, but they are differs by name and their applications. The first one is butyl cyanocrylate. Second one, 4-deoxypyridoxine. Third one, dopamine. Fourth one, isobutyl cyanocrylate. Fifth one, norepinephrine. Sixth one, octopamine as neurotransmitter. Then octopamine as drug. Vanillamine, cyclohexane carbamide. So now we have to identify what we have shown in the particular structure is nothing but a dopamine. Other compounds we are not considering, but they are also having the same empirical formula according to chemistry. Now we have to identify what is dopamine. Dopamine acts as a neurotransmitter. So neural, some of the aged people, they know what is said to be happened in last Sunday. Tuesday or Wednesday, what is said to be happened, they are unable to recollect. And Thursday, they will be very normal. So some 1 or 2 days, what is said to be happened, they cannot recollect due to the neural problems. That is, neural transmission is said to be not properly enabled, so that the continuity of the what is said to be there, the actions that is said to be unable to recollect it from them. So that dopamine can be given over them so that they can easily identify, and it will be act as a neural transmitter. What is dopamine? It is a chemical compound available generally, but according to the normal life, dopamine is nothing but we are having egg. So we are keeping omelette. The egg is converted as omelette for edible purpose. Omelette is said to be very rich in this dopamine. So dopamine omelette, when they are taking omelette, the dopamine content is said to be more. So it will enrich the neuro applications. The nerve cells will be enabled properly. So dopamine is said to be having norepinephrine from storage sites, and they are having beta adrenergic receptors. Based on that, we can have this dopamine as one of the constituent for bio applications. Now we are in slide number 28. So dopamine structure can be classified into different representation. Slide number 28. See, 2-dimensional, we have the same dopamine structure. Ball and stick, that is some representation. Stick only, wireframe, space, ball filled, like that. So based on this, we can identify which application we have. Space ball filled, when van der Waals forces are there, the contraction or expansion can be identified with them. Wireframe, the chemical people, they will draw a single line and they can call it as carbon, hydrogen, like that. That purpose they can be used. 2-dimensional, for the purpose of publications, for the purpose of academic, we are using only 2-dimensional structure. Ball and stick, when you are going for some theoretical study using softwares, the 2-dimensional representation or the 3-dimensional representation using ball and stick is to be mainly considered. Stick only to identify with the hydrogen or without the hydrogen the compound is, and how they are set to be bonded with which type of chain or continuation, like that we can identify. So these are the representation over that. Neurotransmitters, it is said to be mainly by the dopamine. Then human body, slide number 32, it represents some percentage of elements present. Say oxygen 65%, carbon 18%, hydrogen 9.5 like that. Others also less than 1. We are need of sodium 0.2, chlorine 0.2, sulfur 0.3 like that. So when we are going for bio applications, the bio application should lead to a drug or a medicine. It is called a medication. So our material is said to be rich in particular material or not, that we have to identify. Then based on the material what we are having, the composition in our compound or element, then we can go for particular application. As I already told, it is the hydrogen chain or branched chain or side branching in the orthometa para position like that. Next, the functional groups. So we have to identify what are the functional group present in the particular crystal or nanocrystalline materials like hydroxyl, methyl, carbonyl like that. When you're having carbonyl group, they will have more fluorescence applications. N to pi star transition will be more. So it will be excited state values are said to be more. Excitation is said to be more means the emission output will be also more. They'll have good fluorescence or that photoluminescence application. Carbonyl group is there. Carboxyl is nothing but acid. Amino group, phosphate group, they are having some electronic application. It can be used like that. Then alkanes like side number 34. There these different types of functional groups present can be represented over the particular compound or some chemical what we have grown. This can be identified with the help of IR spectroscopy, FT-IR spectroscopy, which functional group present and what is the particular wave number in centimeter inverse can be identified using the FTIR or FT Raman. FTIR is for change in the infrared. Infrared representation is there. So change in dipole moment, and FT Raman is for polarization change, polarization or polarizability change that we can conclude. Functional group with bio impact. See, when you are having these functional groups, that functional groups are mainly for the application of the particular type of interaction possible using FTIR. That functional group will have more application in bio or impact that can be identified. Now, functional groups can be identified with bio impact. As of slide number 35, hydroxyl group, they will have rich found in carbohydrates, so rice and all like that. Carbonyl lipids, so lipids we can identify using fluorescence only. Carboxyl group rich in proteins. Amino group also proteins. Phosphate rich in DNA and ATP, adenosine triphosphate like that. And also common functional groups in biomolecules, aldehyde, carbohydrates, amides like that. These are generally available. And now we are entering into the bio applications. Slide number 38. Slide number 38. So we are having grown one acetoacetonide crystal. This acetoacetonide crystal, they are having identified with some compositions of ketone and other molecules, and they are said to be of second order optical crystals, NLO crystals. Second order NLO crystals, according to physics and chemistry, we can identify. So optically it is active. It is one of the crystals. Now the bio application is confirmed by a software. So it is called molecular harboring. The molecular harboring process of this is identified the post view, and human glucosinide with AAC, according to slide number 38, it represents where it can be projected and where it can be combined like that. So based on the software, we can go for this particular diagram with slide number 38. So this actually we have two main crystals. One is lithium, second one is acetoacetonide. This acetoacetonide, they are having some applications in diabetic, and lithium they are having applications like Alzheimer's disease like that. So now we are going to discuss one by one. This human glucosinide with AACE, that is acetoacetonide crystal, can be identified with the anti-diabetic parameters, that is IC50 value. So the biochemistry, microbiology, biochemistry, biotechnology, they are aware of this IC50. So based on the IC50, we can conclude that it can be act as a drug or not, and what percentage it can be used, what is the toxicity like that. Next, slide number 39 represents the affinity maps of this same AAC crystal. Affinity maps represent grid spacing, say 0.375 for the particular crystal, and we are having hydroxyl group OH with respect to THR65A for human glucosinide, and active sites for the residue balance amount. and we are having minus 5.4 or 5.14 kilocalories per mole. So we can come to the conclusion when our value is more than 5, then it can be act as a good drug. And this here, this is said to be, this crystal is said to be having good diabetic applications. And slide number 40 represents lithium para-nitrophenol crystals for bio applications. So this is for this Alzheimer disease. Those cannot recollect, so it can indirectly act as a neurotransmitter like dopamine. So these crystals. So slide number 41, there it represents, it is minus 5.54 as the value of the binding sites. So I already told, if it is more than 5, then it will be a good receptor for the applications of bio and all, and a good drug value. It will have good tool for the drugs and bio applications. The particular crystal or compound will be. The grid spacing is 0.456 angstrom. Grid is nothing but a like an electronic tool that the spacing will be identified like that. We are having the distance of 2.8, 2.2 and 2.4 Armstrong with the oxygen sites, and the hydrogen bonds and formed between the oxygen sites, that the range is to be. Here it is 5.54 kilocalories per mole. Next, nanometer scales. Now what we have informed is said to be in macro scale. Macro scale is in millimeter, or sometimes it is said to be centimeter. Now nanometer scale, simple. When you are having the bulk crystal, convert the bulk crystal by to nanoscale by ball milling. So we can different types of millings are there. We can go continuously for ball milling of nearly 40 to 45 hours. Then we'll crush the particular macro size into nano size. We can identify for each 10 hour also. Suppose we are running for 40 hours, we can have some 10 grams or 8 grams of the substance. Then some part of the substance can be taken after 10 hour each, and you can identify the particular nanometer scale is said to be there with the SEM or TEM. SEM is for morphology, TEM can be identified with the nano samples. And from powder XRD also, we can get index the values, and we can identify the particular nanoscale using the formulas and all. So what type of nanometer present? The speciality is different, same sample, different nanometer size will have different applications. So same crystal of 40 nanometer or 40 nanometers will not have the same results, but the chemical composition and chemical crystalline nature are same, but the application is differs. That is a specialty of the nano compared to bulk or macro scales. Next represents some of the 43 slide number 43 represents human genetic codes for the elements. So hydrogen, it can be used for protein, bicarbonate and peak control. Hydrogen concentration, it can be used. And for calcium, it can be used for protein, troponin, calmodus, it can be reduced, and signal lines to cells like that. So we can identify that. Slide number 44. Now we are entering into the next level of bio applications. Slide number 44. Here we have taken one crystal. It has to be grown in 2017 and published in 2018. This 4-methoxyphenyl 2-oxo 2H chromine 3-carboxylate. This is one of the crystals that we have grown. 2-oxo 2H chromine, that is nothing but coumarin group. So we have to identify according to chemistry and biochemistry point of view. So 2-oxo 2H chromine, they are used as a coumarin group. So these crystals will have some application based on the hydrogen in some beauty industries, and they will have good anti-diabetic property based on coumarin. And this crystal can be used as an anti-cancer agent based on the coumarin group of combination is to be available, because coumarin group generally they are having good output in anti-cancer, anti-diabetic activities. So here we are identifying 1.279 millimeter as the beam displacement, and we are having 2.9886 microns for NLO utility. So we have to note down one point. So even though we are discussing about bio applications, and the convener of the program is also, she is a microbiology candidate, but the thing is the output we have to relate with some electronic tools also, so that our compound has rich in some other applications we have to identify. So 2.9886 is less than 10 microns. When less than 10 microns, it is a second order NLO, we can conclude. If it is more than 10, we can identify that it is a third order NLO. So third order or second order NLO, non-linear optical property of the crystal can be identified with this. This is one of the industrial applications in electronics because it is a filterability like that. So same applications of the material can be used in pain relieving, hostile to joint pain, mitigating malignant growth, hostile, the anticoagulation property, and they can be used for prescription, beauty care product, and fluorescence colors. And I have already mentioned they can be used for the purpose of anti-diabetic and anti-cancer. So this 45 slide number 45 represent these type of bio applications based on the crystalline nature. They are having the band gap of 3.133 according to the fluorescence or photoluminescence value. The crystal is orthorhombic in nature. So before going into the growth of crystals, we have to identify which type of crystal we are going to grow. So first is orthorhombic or monoclinic. These are the general category. Triclinic, trigonal, these are the rarest category. Hexagonal is very difficult. Cubic is the very basic model like that. So maximum crystals are of three categories only. That one of the categories said to be this type of orthorhombic crystals. Next, slide number 46 represents it is one of the anti-inflammatory agent and anti-pyretic, antiviral, anti-cancer, and anti-coagulation. So anti-diabetic property of this particular crystal already we have done publication, and anti-cancer it is said to be in the pipeline, in the process it is said to be there. So we have to identify based on the chemical composition, physical properties, then we can identify it can be go for bio applications or not. And bio application also, there may be so many change in value. What we are thinking or what we are designing that will not be there as the output because the composition differs, the methodology differs, the solvent beauty of bio applications of the crystal and nanocrystals. Next, slide number 47 represents the ORTOP diagram. So ORTOP diagram is one of the diagrams based on the structural parameters as well as the chemical composition of the particular crystal. We can go for this diagram and we can get it through the single XRD pattern, or we can draw using this softwares, this diagram. This diagram can be used for the purpose of getting the software applications like that. And 48 represents one of the applications of mass spectral LDT and bio applications of D23 DYM crystals. So the crystal parameter is represented in 49 slide number, and that it is belong to triclinic. So one of the triclinic system, and space group is P1 bar like that. So we have to identify ABC parameters for length in three dimension, ABC as X, Y, Z axis, and alpha, beta, gamma for angles, and system triclinic. So we can identify the crystal is said to be properly grown. So slide number 50 represents computational work. So when you're having the boost up of electronic as well as bio applications, then we can go for computationally what we have grown is correct or not. That we can check it out using softwares. The first one is you can call it as unit cell. Unit cell is the basic cell. Repetition of the same will have good crystal structure cell. Next one is particular elemental representation, C3, C4, O2 like that. That representation of the particular unit cell. Then three-dimensional representation is given as the third diagram. So that three-dimensional representation of the D23 DYM. D23 DYM is nothing but diethyl 2,3-oxo dihydrobenzofuran 1-nilidine malonate crystal. So we can call it as simply malonate crystal like that. So different combinations are there. Previously we have X and Y like that, two input only given, and third is said to be the output. When combination of one or two compound is said to be input and third is the output like that. Here more 5 combinations said to be as the reactant, and output will be of one combination that we have to confirm. So we should have some patience. So it will take another 25 days to grow a particular crystal, and for 5 combinations day by day or some 2-3 hours this combination will be there with particular slow processing like that, so that the system or the morphology cannot be varied. It should be a slow process but will have the good yield like that. Slide number 51 represents this molecular potential of the particular crystal. So how the crystal is having electron rich or electron poor. That representation can be identified with this. And slide number 52 represents this solvent of the particular structure of the crystal is said to be there. So solvent representation of the structure. This first one is solvent, second one is solvent with the van der Waal application like that. And slide number 53, weak representation of van der Waal and van der Waal with some 50, more than 50% van der Waal effect that can be identified with this structure. And slide number 54, it is only molecular electrostatic potential. And slide number 55 represents halos effect. Halos is generally for Buddha and other statues, they are having some circular light representation over the head like that. So it can be represented for the wisdom and all. Here halos, before the structure or top of the structure, there is one ring over there to identify or highlight some special representation. That is with the pink color specimen of the structure represents the halos effect. These are all theoretically confirmed using softwares. So many softwares can be there. And so now maximum I have discussed about bio applications. And this slide number 56 represents some industrial applications. So slide number 56, it is a dielectric constant. So some of the people in engineering field and other fields, they'll have this bio applications, that is okay. And next, electronic applications, this dielectric constant is a non-conducting material representation over there, from which we can get whether the material can be applied when you are supplying some electric field. It can be used for inducing the particular energy in a particular field, and for LED lights and photovoltaic device that can be used or not. We can conclude with this. And the slide number 57 represents the fluorescence nature of the particular crystal. So here the first one represents the macro scale, second one represents nanoscale. So many doubts are there and clarifications are there from young researchers. Sir, I have a good crystal. When it's at nano and macro scale, what is the representation? How it differs using spectral line or spectral tool or fluorescence like that. Here both represents the fluorescence spectrum of the particular crystal in macro scale and nanoscale. This macro scale is 407 nanometer, nanoscale is 370 nanometer. So when you are going for nanoscale, the wavelength is said to be decreased, but the energy content is increased because of the smaller size. So wavelength decreased means we are said to be using nanoscale, we can identify, we can go for that. So this bio applications also identified with the help of UV, IR and fluorescence. When you are having the functional groups, particular functional groups, they can identify using the FTIR spectrum. When you are having particular peaks and particular variation in the specimen and any flaw or any broken piece are there found in the particular specimen can be identified with the macro or nanoscale of the fluorescence. And UV, from that it is nothing but a transition, electronic transition. We can identify the absorbent or transmittant nature of the particular crystal. So optical clarity can be identified. When this particular optical clarity is identified for the particular crystal, then we can go for bio or pharma applications definitely based on the specimen nature with this N to pi star transition. I already told carbonyl group or something like that. That also we have to take care. Next, 58, the slide, it represents the photoconductive nature. The specimen, how it allows the light. Now, industrial applications in mechanical nature or mechanical engineering applications. Slide number 59, mechanoelastic tribology. So tribology is nothing but it represents the particular specimen as mechanoelastic in nature. Slide number 60 represents the same. When you are having good nano materials or good crystalline materials, we can grow the material like this helical spring, or when you are having previously the helical spring, we can coat the particular spring using your nano materials or crystalline materials, so that it will have rich in elastic stiffness, rigidness, shear angle like that. Based on that particular angle, it can be rotated. We can identify this can be used for some electric property, mechanical property and optical property like that. when it is rich in optical property, they'll have some applications in the bio. That is the conclusion related to both physics and chemistry. We can combine with the bio applications. Next, the fluorescence study of this crystal represents it is 300 to 600 nanometer variations with 397 as the corresponding peak for the emission. The maximum intensity is due to N to pi star transition. That N to pi star transition nothing but related to carbonyl group, and it is 397 nanometer as the intensity peak for the emission. They are having 3.1255 as the band gap. See, slide number 62 represents the elements present. So we can go for CHNSO studies to identify the combination of elements present over the particular macro or nanoscale. So this can be equivalent to FTIR. FTIR, their functional groups are present. Here the elements percentage can be identified. So 100% what we are having experimentally and theoretically, that we can identify using this CHNSO experimental and theoretical calculations for the elements present in the particular crystal. This photoconductivity of the particular specimen is identified with 63. This dark current and photocurrent, like that we can identify. This is a negative photoconductive nature, like that. 64 represents antidiabetic activity of picolinium malate. So we have grown one crystal. Picolinic acid and malic acid is taken as 1:1 ratio. It shows AD action. AD is nothing but antidiabetic action. So it is because of the triazole group, due to closeness of triazole and alanine group in the molecule. So picolinium malate is similar to that of these groups, and picolinic identification represents IC50 as 20.26 from the table we can identify. So next slide 65 represents the antidiabetic nature with concentration and inhibition. There 20.26 as the IC50 value for this PM crystal, and when it is said to be nanoscale, it is 20.2 only. So 0.06 variations are there. Whenever you are having IC50 value reduced, your material is very good for bio and it can be used for some medical purpose, like that. So IC50 value should be very reduced one, so that it will have some impact on biological and medical. So this particular graph shows concentration and inhibition, and it varies for the particular PM crystal. PM nano, next slide, there it is 20.2, previously 20.26, here 20.2. That means 0.06 variation in the macro and nano scale. Same concentration and inhibition. So converting the macro crystal to nano, the variations are said to be observed. This is already published with the help of the HOD, the microbiology. She has helped in confining the antidiabetic activity for the particular crystal of PM. This is published in elsewhere. And next slide represents AAP and BM. There we have compared the macro and nanoscale directly. Macro is more 31.03 and nano is 29.01. So nano is best suited for antidiabetic compared to nano. Macro, this 29.01 is the best suited nano value, and then it can be adapted for antidiabetic in a fine manner, like that. So based on the concentration, the variations will be there. The percentage of inhibition to the type of crystals. In the crystal, they are having some antidiabetic value as 26.28 in macro line. So these are the applications of macro and nano. And slide number 73, that represents temperature dependence of dielectric constant for the particular crystal. Slide number 73. And the remaining is said to be some electronic applications. I am not discussing that. Here, the dielectric constant and temperature is said to be identified with the variation in the frequency. So these 4 graphs represents 4 or 5 graphs. It represents this particular 100 hertz to 1 megahertz variations for this. So dielectric constant, it is the tool from which you can identify it can be used for LED or not. LED we need of some optical property. When the material is optically rich, it can have some good bio applications. Optically good means it have transmittance is said to be good, and the transmittance is very good. It will have transmittance good means it is said to be of high pure. We are having pure specimen, like that, when the transmittance is said to be more. So it have it definitely it will have some bio applications or pharma applications. These are the representation over this particular 73 and 74 slides, and directly I will go to slide number 86. Slide number 86. Here the antidiabetic activity of the particular crystal is said to be given. That crystal is having IC50 value as 37.5 and 30.4 for bisglycine lithium bromide crystals for macro and nano scales, from which we can identify these crystals are best suited for anti-diabetic only when they are said to be present in nanoscale. So that is the value given in slide number 87, and from which we can come to the conclusion that glycine and bromide are not due to the lithium over there. This is the conclusion from the slide number 88. So when you are having the lithium, glycine, bromide, that crystal, bromide is said to be somewhat harmful. When it's said to be in nanoscale, it will act as a biotool. That is the property. And glycine is said to be having good anti-diabetic. That is, we are having good literature survey from 1980s also. So lithium, it can be used for batteries like that. So lithium will not have that much impact over anti-diabetic property for the particular crystal. Glycine and bromide, they are having present over the particular crystal for anti-diabetic property. Next, slide number 89. So this represents the type of growth of this crystal and the nanoscaling. So ABC alpha beta gamma represents this particular type of crystal is belong to monoclinic. There for first 15 hours we are having 61 nanometers, and for 30 hours we have 43 nanometer. After 40 hours we have 34 nanometer. After 45 hours we are having 22 nanometer. That we have not mentioned here. It is the work done after this particular preparation of this. So this lithium bromide BGLBM crystals, they are having macro scaling of 13, 7 and 5 millimeter cube values. They are very rich, and we can go for the particular bio application, anti-diabetic application, that already we have discussed the IC50 values. Next, the same crystals having some electronic applications based on the micron size, that is given in page number slide number 90. That they are calling it as influx. Influx is for the purpose of filter tools. Filter means unwanted light can be filtered over there, or unwanted wave can be filtered like that. So filter applications 2.4 for nano. So we can it can be best suited for some NLO applications, we can conclude. Anti-diabetic property is 37.5 and 30.4 according to slide number 91. So that is the best suited for anti-diabetic case. And anti-inflammatory also, the studies are not yet, we are sent for anti-crystals. Slide number 92 represents the crystal size and powder XRD. So most of them, they are having the doubt, either physics, chemistry or biochemistry students, they are having, sir, we are taking the single XRD parameters A, B, C, alpha, beta, gamma. What is the purpose of taking the powder XRD? Single XRD is the elemental representation or the particular crystalline representation lattice parameters A, B, C, alpha, beta, gamma values only. So counter verify that. To counter verify, you have good nano sample, you have to go for powder XRD for intensity versus 2 theta, like Bragg applications, 2D sin theta equal to n lambda, like that, from which you can go for the identification of the nanoscale using the formula like that. So here it is said to be indexed. When you are having only graph, it is called powder XRD pattern. Here it is indexed with 1, 4, minus 2 like that. It is from indices, Miller indices. When Miller indices is nothing but we have ABC values for a particular case. We have to take 1 over there value, 1 by value, take common LCM, from which we can identify that best suited for some citing this value or indexing this value, so that we can go for some other applications, or crystallinity, the purity of the crystal can be identified. And also for nano applications, we are in need of same TEM as well as this particular type of powder XRD pattern. Next sample with the hours, that is going to be given in slide number 93. The fluorescence in the case of BGLBM is given in slide number 94. See here it is 403 nanometer according to slide number 94. It is 403 for the case of macro, and according to slide number 95 for nano it is 397. So some 6 nanometers to be varied for macro and nano. And this macrocrystalline will have some inhibition concentration value, and nanocrystal will have some inhibition concentration value according to 96 slide, so that how the variation of IC50 is said to be obtained, like that from graph we can identify. So no need to go for literature survey or the data. From the graph we can identify the nano is the best and macro is somewhat comparatively best compared equivalent to nano, but it's not suited best as of nano, like that. So we can conclude with the diagram itself. Next, anti-diabetic for the elements, it is already discussed. Zinc oxide anti-diabetic, titanium and copper oxide, and this type of some of the crystals they are having anti-diabetic. So when our material is not having anti-diabetic applications, it will not show any effect over the IC50 as well as the inhibition rate. When you are giving the concentration, the inhibition values are said to be nearly zero or very, very low value. That indicates that our material will not have anti-diabetic applications. And when you are having anti-diabetic, we should go for anti-cancer and other type of applications like anti-pyruvitic, anti-larval, antibacterial, like that. Antibacterial generally they are doing for all set of nano samples like zinc oxide or other type of oxide coating, like that. Here this anti-cancer, when you are having pyron, benzopyron, like that, definitely you will have anti-cancer applications for the particular crystals you have. And coumarin group, I already told, 2-oxo 2H chromine group, that is called coumarin group, will have anti-diabetic and anti-cancer activity. Direct iron, not iron, direct iron it will not be considered. Ferrous derivatives, so some application with the ferrous, they will have anti-cancer application. Acetoacetonide, one crystals I told, that crystals anti-diabetic we have completed. That acetone or acetone derivatives, acetoacetonide crystals, they have anti-cancer activity. So based on the structural parameters, based on the previous work, and based on our combination of the crystal with anti-inflammatory and anti-diabetic, we can come for the conclusion for the next application like anti-cancer, like that. So these are the materials, they are best suited for anti-cancer, that we can choose and we can go for that. Next, physio or pharma. Generally bioimpact is said to be directly given. For pharma application, it should be act as a drug. Physio means here tapping means when someone is or some hand somewhere. So for the tapping we are need of some nanomaterials or nanocrystal materials, like that. And also tapping tools, we are applying some high frequency of more than 20,000 hertz, that is high frequency generator application. These are all nothing but physics applications and other chemical applications, like that. So this nanomaterial can be used as a tapping tool or joint tool, and high frequency can be applied over the tapping. Different type of tapping are there, the physiotherapists they know very well. So high frequency is said to be ultrasonic frequency. It is from physics or chemistry, from lab we can have that ultrasonic interferometers to get that particular frequency. And upon conclusion, slide number 99 represent all the crystals are grown by slow evaporation solution growth method, and they are having orthorhombic or monoclinic system. Dielectric constant or dielectric loss we are having. The anti-diabetic property can be analyzed with IC50. Negative photochemical nature can be identified. This is the general information about the conclusion of the particular specimen and all related to all specimen. Overall conclusion, it is given with 100, slide number 100. Good quality enamel crystals are grown. So non-linear optical crystals converted to nanoscale. It is by milling method. So slide number 100, converted to nanoscale by milling, and we are having anti-diabetic, anti-inflammatory work that we have substituted, and anti-cancer activity we have to substantiate the same with the data that we will give the same soon. The electronic fluorescence and filter applications, as of slide number 100, it represents good FL and filter and influx applications. Qualitative and quantitative measurements can be given over there with the values of quantity of specimen and quality of the output that represents over here. Bio and industrial utility. Bio and pharma utility is one channel. Industrial utility by electronic or mechanical, it is said to be other channel. So comparison of macro and nano scaling is said to be there to identify our specimen is said to be good or not. So biopharma is biological and pharmaceutical or medicinal applications like that. And industrial, I have given two representation: fluorescence and influx. This electronic tool, and mechano-elastic applications like tribology. You can go for industrial application of the specimen. So we have concluded with 4 applications of the particular specimen, same crystal or nanocrystal. One is bio, one is pharma, one is electronic, one is mechanical tool. Like that, all we have concluded with the same specimen. So some of the journals what we have published, I am giving this for the reference as one of the references for the publication, so that we can get to the conclusion that what kind of work we can go with the sample what we have grown in our particular lab, or some synthesizing with this commercial application. Either we can get through the commercial values like that. So slide number 102 represents our some of the publication in our R&D. So we are having 200 UGC we have published, UGC listed journal, 100 plus UGC CAR listed we have published, 43 papers in elsewhere and Springer we have, and the list of publication in 2019 December to till date with anti-inflammatory hardness characterization of some of the crystals, CPD crystal. With the help of Madam Flora, we have concluded this paper. Here it is published in elsewhere. So anti-inflammatory agent and anti-diabetic agent, this particular crystal, and anti-cancer we have not yet studied for this particular crystal. So here the crystalline hardness, so hardness value is said to be found for the particular crystal. The inflammatory nature of the crystal also studied. And next one, here computational analysis of dementia for Alzheimer disease of lithium paranitrophenol. I already told lithium is a Alzheimer disease representation. And next slide, we are having some bio applications. And next slide, we have hardness and anti-diabetic applications of LEAF crystal, slide number 105. And growth and dielectric properties, slide number 106. And theoretical applications and growth and characterization, slide number 107. These are published in AIP, American Institute of Physics journal. And slide number 108, this just an acceptance of anti-diabetic and aluminum malate crystals as an anti-diabetic agent is accepted in elsewhere. And slide number 109, this LVP, L-valenium picrate as an anti-diabetic agent accepted in elsewhere. And here, so this one important point I would like to discuss in slide number 110, that we have given this first author for the particular scholar who has worked over there. This we have used the domestic oven to synthesis ZNO particles from neem. There it can be used for anti-diabetic agent that we have not yet published that paper, and here it can be universal fabrication. So slide number 110 represents Springer journal. There we are having appreciable size solar cell utilization of the particular Azardica indica leaves by domestic microwave oven. So oven we can use it to synthesize the same that can be applicable for solar cell, and we are having anti-diabetic, anti-inflammatory also that we have done. It is said to be in the pipeline to publish, and anti-cancer we have sent the sample for the study. So from the same effect we will have some bio electronic mechanical applications that you can conclude with this. And this is published in Springer with the Journal of Materials in Electronics. It is having 2.2 impact factor. In February 2020, it was published. End of January, it is published. And we are having other publications like PayDemo and Web of Science journals that what are the bio applications we are having like that. And slide number 116. Wish you all the best for the faculties as well as scholars and other people who are present over here. And slide number 117 for your patience hearing for the lecture what I have given. Thank you. And slide number 118. If you have any queries, please ask.
Thank you, sir. You gave a nice presentation. It is very informative and educative one. Participant posed some questions for their clarification, sir. Shall I convey the question, sir?
Yeah, no problem, ma'am.
First question is, what is correlation between IC50 and toxicity?
Yeah, IC50 is the inhibition concentration value. When the values are said to be very low, then it is best suited for bio applications like that. Directly, we cannot identify the toxicity from this, but we can go for toxic studies separately. But here, when the values are very less, then we'll have more application as a drug. When commercially what we are having the compound or crystal or material or nanoparticles like that, when it's said to be a drug, then it can be used as an bio element like that. Otherwise, it is only a crystal or an physics or chemistry materials like that. So IC50 value can identify the particular material as an one of the drugs for anti-diabetic or anti-inflammatory like that. So toxicity will be maximum, it is reduced. If you want really what your sample is having, how much percentage of toxicity nearly with respect to the particular values or like that, we have to go for toxicity of the particular specimen study separately. So this will not give the toxicity directly, but some IC50 value, less values will have more impact on drugs. That only we can conclude. Directly we cannot give toxicity value based on IC50.
Thank you, sir. Nicely clarified. The next question is it possible to predict the biological activities with the FTIR data, sir? Sir?
No, I'm not clear with the particular question. Repeat the question. Repeat the
sir.
question. Signal problem. Repeat the question.
Is it possible to predict the biological activities with the FTIR data?
No, we can get the information. This functional group is present like that. So suppose you are having chromium or coumarin or biron like that, we can identify this crystal or particular nanomaterial can be used for some bio applications like that. 100% we cannot confirm because of the change in chain, the property vary. Change in the dimension, the property vary. Change in the length as of nanometer scale, the property vary. So we can conclude when the pyran is present, we can identify that it can be used for anti-cancer. And really when you are giving for anti-cancer only, you will get the proper result. It is one of the tools to get the information that it can be used for the particular type like that. Directly, we cannot conclude anything from FTIR and all. FTIR is the function group present in the element. It's one of the tools to identify. We can go for next step.
Thank you, sir. The next question is, what are the softwares used for computation and analysis of crystals? Is there any free online software available?
Yeah, free softwares are there, but that much not said to be successful. As of my knowledge is concerned, we are having Gaussian software. We are having other software also, software tools also. We can go for based on your chain, based on your number like that. So before that, we have to go the geometry of the particular crystal. We should go for some analysis and the freedom we have to identify. What are the type of freedom over there? If it is a linear chain, then the formula differs. If it is a non-linear chain, the formula differs. Based on that, we can go. And the molecule is said to be having more atoms, biological application and your the theoretical or computational or software applications said to be get deferred. So when you're having less atoms, then we can go for fine tuning of the atoms to confirm the XRD pattern or any other tools like a higher energy gap of HOMO, LUMO and other applications like that. Generally, we are using Gaussian softwares and some of the other tools also we are trying. These diagrams, what I have shown is with respect to JavaScript we are having. That we are not having, our circle having, that we are utilizing the JavaScript to identify the particular tools for the particular specimen. It will not be confined for all the specimen. We have to analyze for the particular structure what we are having. Then only we can go for software applications.
Thank you, sir. With your knowledge, shall we move to the valedictory of this session, sir?
No problem, ma'am. You can.
Thank you, sir. Now, I invite Mrs. A. Lakshmi, Organizing Secretary, Assistant Professor in Microbiology Department, to deliver a vote of thanks.
A very good evening to all. Sunshine is a welcome thing. It brings a lot of brightness. With these words, I would like to thank Almighty for the success of today's session. Yesterday is not ours to recover, but tomorrow is ours to win or lose. According to the quote, it's my privilege to thank our principal, Reverend Sister Dr. Eugene Namala, for her support. It's my pleasure to thank our college secretary, Reverend Mother Claudine Orokia Mary, for her kind and polite guidance. I would like to thank our college administrator, Reverend Sister Antonia Mall, and our Vice Principal, Reverend Sister Dr. Vimali Mary, for their valuable inspiration. Satisfaction lies in the effort, not in the attainment. With the quote of Mahatma Gandhi, I would like to extend my sincere thanks to our Honorable Chief Guest, Dr. K. Senthil Kannan sir, for his insightful information about biopharma and industrial applications of crystalline materials. Thank you so much, sir. The speed of the boss is the speed of the team. By Lee, yes, the perfect person who is none other than our department HOD, Dr. R. Krishnaveni ma'am, for her guidance and support. Thank you, ma'am. Great things never done by one person, they are done by a team. With this quotation, I want to extend my gratitude to our participants, includes professors, assistant professors, associate professors of different colleges and universities from various disciplines. I thank you one and all. I would like to thank our comparer, Dr. M. Nitya ma'am. Thank you, ma'am, for your comparing. It's my pleasure to thank our college professor, assistant professor, Mr. Magesh sir, for his good effort to get success of today's international FDP as a technician. Once again, I thank you one and all. Thank you, ma'am.